Alright, so today we're going to talk about cell transport, and more specifically we're going to talk about passive and active transport. Make sure that you're following along in your textbook for this video. There's a lot of vocabulary, and make sure that you're reading and understanding all the terminology that we're going to use throughout this lesson. Alright, so there are two types of transport. You have active transport and passive transport. Let's start with passive transport. Passive transport is a type of transport that does not require energy. And the type of cell energy that we use is ATP, and we're going to talk about that in a little bit more detail later. This is typically moving from a high concentration to a low concentration. So another way of saying that is down a concentration gradient, going from high to low. The opposite of that is active transport. Active transport is the type of transport that uses ATP, and it goes against the concentration gradient. So what that means is moving from an area of low concentration to an area of high concentration. Over here is a sample diagram of what active and passive looks like. Passive, we're going to talk about some specific examples, but active transport here, the key part is the use of ATP. So there are two main types of passive transport, and that's going to be simple and facilitated diffusion. Simple diffusion is going to be moving small nonpolar uncharged molecules across the cell membrane. Remember that cell membrane is selectively permeable, and it also has a couple characteristics like a hydrophilic and a hydrophobic end. So these small nonpolar and uncharged molecules that can easily pass through the cell membrane are soluble in the hydrophobic interior, so where those fatty acid tails are touching each other. Some examples of molecules that will undergo simple diffusion are oxygen and carbon dioxide. The flip side of that, or facilitated diffusion, is going to be for large polar and charged molecules because they're not very soluble in that hydrophobic interior or where those fatty acids are touching each other. And there are two main types of proteins that are used in facilitated diffusion. The first is what's called a channel protein, and these are those proteins that go across the entire membrane of the plasma membrane. And there are two specific types of channel proteins. One is what we know as an ion-gated channel, and this is a channel protein that is activated by a ligand, and the ligand is a chemical signal that attaches to the protein, and it causes that protein to change shape, moving a molecule across the plasma membrane. I'll show you a picture of that in just a second. We also have aquaporins, and these are protein channels that carry water across the membrane. So water is pretty large, and these aquaporins allow that water molecule to move quicker, or larger amounts to move quicker. We also have carrier proteins, and these are proteins that change shape and release the molecules on the other side. Again, I'm going to show you a picture of that in just a second. So here's what simple and facilitated diffusion looks like. Simple is just small molecules moving through the plasma membrane without a protein. Then we have facilitated diffusion through a non-specific transporter. That just means it's a protein that has an opening and all different types of molecules can move through it. Then we have one with a specific transporter. And so here you can see some examples of a carrier or a channel protein. They actually change shape and they move the molecule across the cell membrane. So here it's open at the top, and down here it's open at the bottom. That's a change in the shape. And then of course we have osmosis, which is the movement of water. And that's using those aquaporins, which is also a form of facilitated diffusion. So I just started talking about osmosis and how that's the movement of water. There's a lot of terminology that goes on with osmosis. And the first is this idea of osmotic pressure. This is the pressure needed in a solution to prevent the flow of water across a membrane by osmosis. And when we're talking about solutions or comparing two solutions, we have three terms that we use. The first is hypertonic. A hypertonic solution has a high solute concentration compared to whatever you're talking about. The hypotonic solution is the one with the lower solute concentration. And an isotonic solution is when we have two solutions that are equal in solute concentration. So remember, the solute is the substance that's being dissolved in a liquid. And this has major impacts for plants and animals. In an isotonic situation, these cells are going to be pretty ideal. And remember also, plant cells have a cell wall, which is an extra outer layer surrounding their cell membrane. A less than ideal situation is in a hypotonic solution. This is a low solute concentration, and this can actually allow water to move into the cells, causing in animal cells those cells to lyse or to burst. And then in plant cells, it actually produces something that's called a turgid pressure. And this turgor pressure is the pressure that is exerted because there's an extra cell wall. So plant cells can't easily burst like these animal cells here. And we're talking about red blood cells in particular. We can also have these in a hypertonic solution, which is when there's a high solute concentration. And water is going to want to move out of the cells, and it can cause those cells to actually shrivel up. And the same with plant cells. And we call this plasmolysis. And that's when the cell membrane actually pulls away from that cell wall. So there's a lot of terminology here when we're talking about these different types of solutions. So a little bit more detail about active transport. There are two main types. We have primary active transport and secondary active transport. Primary active transport is driven by the hydrolysis of ATP. So that's splitting ATP into what's known as ADP and a phosphate. When that third phosphate is released, it releases a ton of energy. And so an example of a system in your body that uses primary active transport is the sodium-potassium pump. This is going to be a pump that allows the concentration gradient of sodium and potassium to build up, which drives a lot of important processes. And we're going to talk about that a little bit later on in the year. An example of secondary active transport is not using ATP directly. In other words, it's going to use the concentration gradient that was built due to the use of ATP, and that's going to actually drive secondary active transport. So an example would be the concentration gradient that was set up from the primary sodium-potassium pump allows the influx of glucose into cells. So that's what actually regulates the amount of glucose that we need within all of our cells. So it plays a really important role in kind of playing off of the primary active transport. Two more types of active transport. You have exocytosis and endocytosis. Exocytosis is when materials are secreted from the cell in the form of vesicles. And most of the time those vesicles are pinched off from the Golgi apparatus following that endomembrane system that we talked about with cells. And then those materials are expelled from the cell. The opposite of that is endocytosis, and this is when materials are brought into the cell, and there are three main types. You have phagocytosis, which is also known as cellular eating, and that's going to be bringing in actually particles or even smaller cells into a cell. We have penocytosis, which is cellular drinking, or the bringing in of fluid into a cell. And then we also have receptor endocytosis, and this is very similar to phagocytosis. The only difference is that there's a coated vesicle that forms around the materials that are being brought in. So I would like you guys to actually read in your book the section on receptor endocytosis and learn about those different vesicles and come to class prepared to talk about them. All right, guys, have a good night, and I will see you tomorrow.